Before a single result reaches a report, a technologist has usually done math, dilutions, corrections, ratios. Get the formula right and the result is trustworthy. Get it wrong and a perfectly good instrument gives a perfectly wrong answer. This is the complete toolkit. Start with the single most used formula in any lab, the dilution equation. Concentration 1 times volume 1 equals concentration 2 times volume 2. It holds because the total amount of solute never changes, only how much liquid it's spread through. Worked example, you have a 500 milligrams or DL glucose stock standard and need 100 milliliters of a 100 milligrams or DL working standard. Solve for V1. V1 equals C2 times V2 divided by C1, which is 100 times 100 divided by 500 to 20 milliliters of stock, topped up with 80 milliliters of diluent. The dilution factor itself is simply total final volume divided by the volume of original sample used, expressed as a ratio like 1 to 10, meaning 1 part sample in a total of 10 parts. Serial dilutions repeat this doubling process in sequence. Each tube becomes the source for the next, having concentration every step. 1 to 2, then 1 to 4, then 1 to 8, then 1 to 16, commonly used for antibody titers and endpoint detection. If a titer reads positive up through the 1 to 64 tube and negative at 1 to 128, the reportable titer is 64, the last dilution still showing a positive reaction, not the first negative one. A genuinely common bench error. Reporting a result and forgetting to multiply back by the dilution factor. A sample diluted 1 to 10 that reads 25 on the instrument is actually 250 in the original specimen, not 25. Every other formula in this video eventually touches a diluted sample somewhere. Get this one automatic and everything downstream gets more reliable. Move to hematology, where 3 formulas derived from a basic CBC describe the size and hemoglobin content of an average red blood cell, and immediately narrow down the anemia differential. Mean corpuscular volume is the average size of a single red cell, hematocrit percentage multiplied by 10, divided by the red cell count in millions per microliter, giving a result in femtoliters. Mean corpuscular hemoglobin is the average mass of hemoglobin packed inside one red cell, hemoglobin concentration times 10, divided by red cell count, giving a result in picograms. Mean corpuscular hemoglobin concentration measures how densely packed that hemoglobin is relative to the cell's own volume, hemoglobin divided by hematocrit times 100, giving a percentage or equivalent G or DL value. Run all 3 off one real CBC. Hematocrit 45%, hemoglobin 15 G or DL, RBC count 5.0 million per microliter. MCV comes out to exactly 90 femtoliters, MCH to 30 picograms, and MCHC to 33.3%, a textbook normal red cell. These 3 numbers instantly classify an anemia. MCV sorts microcytic from normocytic from macrocytic, while MCHC sorts hypochromic from normochromic. Together they point a technologist toward the likely cause before any further workup. One more index worth knowing, RDW, red cell distribution width, measures how much red cell sizes vary from each other. A high RDW with a low MCV, for instance, points specifically toward iron deficiency over thalassemia trait. 3 simple divisions off one CBC, and a technologist already knows the shape and color of the anemia before a single additional test is ordered. 2 more hematology formulas fix a specific, well-known measurement problem. Automated counters that mistake other cells for white cells, and respiratory percentages that are meaningless without knowing the actual red cell count behind them. Automated analyzers count nucleated red blood cells alongside true white cells, artificially inflating the WBC count. The correction, WBC times 100, divided by 100 plus the number of NRBCs counted per 100 white cells. Worked example. An automated count reads 15,000 white cells, with 25 nucleated red cells seen per 100 white cells on the differential. Corrected WBC equals 15,000 times 100, divided by 125, exactly 12,000, a full 20% lower than the raw count. The second correction. Respiratory percentage alone is misleading in anemia because a shrinking total red cell pool can make the percentage look falsely reassuring. The absolute respiratory count fixes this, retic percentage divided by 100, multiplied by the actual red cell count. Worked example. A respiratory count of 2% against a red cell count of 4.5 million per microliter, gives an absolute respiratory count of 90,000 per microliter, above the normal 25,000 to 75,000 range, indicating genuine compensatory marrow response. The trap both corrections avoid is the same one. A raw percentage or raw count can look normal or look alarming, purely because of what else is happening to the total cell population. Always correct back to an absolute number before interpreting. 2 corrections, 1 shared lesson. A raw number often analyzer is a starting point, not a final answer until it's corrected for what else is going on in the sample. Move into clinical chemistry, where a handful of formulas built entirely from a basic metabolic panel do genuinely heavy diagnostic lifting, starting with one of the most clinically important calculations in the entire lab. The anion gap estimates unmeasured negatively charged particles in plasma sodium, minus the sum of chloride and bicarbonate. Since total charge must balance, a gap larger than normal means something else is generating negative charge. Worked example: sodium 140, chloride 95, bicarbonate 10. Anion gap equals 140 minus 105, 35. Dramatically elevated, consistent with a high anion gap metabolic acidosis like diabetic ketoacidosis or lactic acidosis. Corrected calcium adjusts a total calcium result for low albumin, since roughly 40% of calcium travels bound to albumin. A low albumin makes total calcium look falsely low even when the physiologically active unbound fraction is genuinely normal. Worked example: measured calcium 7.8 mg or dl, albumin 2.0 g or dl. Corrected calcium equals 7.8 plus 0.8 times 2. Exactly 9.4 mg or dl, comfortably within the normal 8.5 to 10.5 range once the low albumin is accounted for. Calculated osmolality estimates plasma osmolality from routine chemistry values alone, twice sodium plus glucose divided by 18, plus BUN divided by 2.8, each division converting mg or dl into the millimolar units osmolality actually needs. The real diagnostic value comes from the osmolar gap, measured osmolality from an osmometer minus the calculated value. A gap over 10 suggests unmeasured osmotically active substances, classically toxic alcohols like methanol or ethylene glycol.
Quick pause: if this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming.
3 formulas, 1 basic metabolic panel, and 3 genuinely different diagnostic questions answered. That's the real power of clinical chemistry math. 2 more formulas that clinicians lean on constantly, both estimating something the lab can't measure directly: how well the kidneys are filtering, and how much LDL cholesterol is actually present. Cockcroft-Gault estimates creatinine clearance from a single serum creatinine, without needing a full 24-hour urine collection, 140 minus age, times weight in kilograms, divided by 72 times serum creatinine, multiplied by 0.85 for female patients. Worked example: a 60-year-old male, 70 kilograms, serum creatinine 1.2 milligrams or DL. Clearance equals 140 minus 60 times 70, divided by 72 times 1.2, working out to roughly 65 milliliters per minute, mildly reduced from a normal of around 90 to 120. Modern practice increasingly reports eGFR via the CKD-EPI equation instead, more complex, incorporating race-neutral coefficients and better accuracy across a wider range of kidney function, always software calculated rather than done by hand. The Friedewald equation, meanwhile, calculates LDL cholesterol without ever measuring it directly: total cholesterol minus HDL minus triglycerides divided by 5, that last term estimating VLDL cholesterol from its roughly fixed triglyceride ratio. Its one hard limitation is guaranteed to come up on the bench or on an exam. Friedewald becomes unreliable once triglycerides exceed 400 milligrams or DL. Since the fixed TG to cholesterol ratio assumption no longer holds, direct LDL measurement is required instead. 2 formulas, 2 organs worth of information, neither requiring an invasive test. That's exactly why both remain in daily use decades after they were first derived. 2 very different lab sections, coagulation and microbiology, each lean on one formula that solves the exact same underlying problem, making results comparable across different reagents, instruments, and labs. Prothrombin time varies significantly between labs because thromboplastin reagents differ in sensitivity. INR standardizes this, the patient's PT divided by the mean normal PT, raised to the power of the reagent's own International Sensitivity Index. Worked example. A patient PT of 24 seconds against a mean normal PT of 12 seconds with a reagent ISI of exactly 1.0. NR equals 24 over 12, raised to the power of 1, a clean 2.0, squarely within the standard therapeutic anticoagulation range of 2.0 to 3.0. ISI matters because without it, the same patient could get a meaningfully different PT ratio on two different reagents. INR exists specifically so a 2.0 means the same thing whether it's measured in Delhi or Denver. Switch to microbiology, where colony-forming units per milliliter quantifies bacterial growth from a plated culture. Colony count, multiplied by the dilution factor, divided by the volume of specimen actually plated. Worked example, 150 colonies counted from a 1 to 1000 dilution, with 0.1 milliliters plated. CFU per ml equals 150 times 1000, divided by 0.1, 1.5 million CFU per ml, well above the 100,000 threshold typically defining significant bacteriuria. One standardizes a clotting result across the entire world. The other turns a handful of visible colonies into a clinically meaningful concentration. Both are exactly why raw counts alone are never the final answer. Step back from individual patient results to the statistics that judge whether the lab itself is performing correctly, the formulas behind every internal quality control chart. Coefficient of variation expresses imprecision as a percentage, independent of the units or magnitude of the analyte. Standard deviation, divided by the mean times 100, making precision comparable across completely different tests. Worked example. A QC pool with a mean of 100 mg or DL and a standard deviation of 2.5 mg or DL gives a CV of exactly 2.5%. Comfortably within the under 5% target most chemistry analytes are held to. Sigma metric goes further, expressing overall assay quality as a single number. The allowable total error minus the observed bias, divided by the CV. Higher sigma means fewer errors reach a patient result before detection. Worked example. An assay with 10% total allowable error, 1% observed bias, and 2% CV scores a sigma of 4.5, solidly good performance, though short of the coveted 6 sigma world-class benchmark used in 6 sigma quality frameworks. The everyday reference range itself is a statistical construct too, typically the mean of a healthy population plus or minus 2 standard deviations, capturing roughly 95% of that healthy group by definition, not a hard line between sick and well. CLIA and most accreditation bodies expect labs to hold CV under roughly 5 to 10% for most chemistry analytes and even tighter for critical tests like troponin. Exceeding target CV is exactly the kind of finding that should trigger a root cause investigation, not just a repeat run. Every patient result on every report is only as trustworthy as the CV and sigma metric sitting quietly behind it. This is the math that makes trusting a lab result reasonable in the first place. One last foundational formula worth a mention. Nearly every colorimetric assay you'll ever run reduces to Beer-Lambert's law. Absorbance equals molar absorptivity times concentration times path length. Every glucose, every creatinine, every liver panel value ultimately traces back to this. Let's put several formulas to work on one real bench scenario. A urine culture reads 150 colonies from a 1:1000 dilution. That's 1.5 million CFU or ml, clearly significant. The same patient's chemistry panel shows a corrected calcium of 9.4, an anion gap of 14, and a Cockcroft-Gault clearance of 58 milliliters or min, a coherent clinical picture assembled entirely from formulas. Quick check before we close. A serum triglyceride level comes back at 550 milligrams or DL on a patient's lipid panel. Which formula from this video should not be used to calculate their LDL cholesterol? Carry these numbers back to the bench. Dilution factor equals total volume over sample volume. Friedewald's ceiling sits at triglycerides of 400. A CV under 5% is the everyday chemistry target, and a sigma of 6 remains the world-class benchmark. Dilutions, red cell indices, corrections, electrolytes, kidney function, coagulation, colony counts, and QC statistics. Every one of them is just arithmetic with a clinical meaning attached, and that meaning is exactly what makes a technologist more than a button pusher.
Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.